Welcome back, folks, to our continuation of our discussion of invertebrate diversity. This is part 2 of our exploration of invertebrate diversity, and so we want to pick it up with the lophotrochozoans. So what's unique about the lophotrochozoans is they are bilaterian, so they have bilateral symmetry, and they do have triploblastic development, so they have 3 germ layers. But one thing you want to pay attention to here is about their coelom and digestive tract design. They have a coelom, and the digestive tract is considered to be a complete or an alimentary canal because it has 2 openings, a mouth and an anus. And we'll see that this clade includes the lophotrochozoans, the ecdysozoans, and the deuterostomia, all belonging to the bilateria. So we can see that these 3 groupings here, lophotrochozoans, ecdysozoans here, and deuterostomia, all belong to the bilateria. First, let's take a look at the flatworms that are part of the lophotrochozoans. These are the platyhelminths here. So they live in all kinds of habitats, marine freshwater, damp soil. Many are parasites, such as flukes and tapeworms. Unlike most triploblasts, flatworms are acoelomates, and that's pretty important. Remember previously we talked about every time you see the letter A in front of a word, it means without. So this means it's without a true coelom. They don't have a body cavity because they're filled with mesoderm. And so flatworms have a gastrovascular cavity with only 1 opening, so they have a GVC. You'll see later on they have a specialized kidney called a protonephridia that's involved in excretion of their nitrogenous waste. They have a unique dorsal ventrally flattened body, and that's an advantage because it makes all the cells in close contact with their environment, which is water in this case, or moisture. So they don't need complex transport systems. They can do exchange right through their body surface. And then they'll have specialized folding structures to maximize surface area to increase the ability to do gas exchange. Here's an example of that, comparing the idea of maximizing surface area. So you can see up here that you've got a cube, and the cube has a volume of 27 cubic centimeters. And then you have this wafer, rectangular wafer, that also has a volume of 27 cubic centimeters. But in this case, the surface area is much greater. It has 78 square centimeters surface area as opposed to 54 square centimeters here. So flattening is an advantage for increasing your surface area to volume ratio, which is better for exchange. And these are all pictures of different ways animals increase their surface area by folding, projections, branching, flattening. And you'll see that through all the rest of the animal kingdom. So planaria is one example of a platyhelminth, or Dugesia that you might study in lab. They have a more complex nervous system than cnidarians. They are hermaphrodites. They can reproduce asexually or sexually. And in lab, you'll study the functions of these unique organs here. The pharynx, which is what they use to suck up their food, excreting digestive enzymes through the pharynx. It protrudes out the mouth here. And then they also have a unique GVC because it's branched. As you see, one branch here, and then another branch here, and then another branch here. And then each of these branches has folds in it to increase the surface area for better absorption. They also have ventral nerve cords that are solid, and they have concentration of ganglia at one end, which we learned with cephalization. There are eye spots there, which are light-sensitive organs, but they're not real eyes. Another group of platyhelminths that we need to look at are the trematoda, or trematodes. They're usually parasites, and they alternate sexual and asexual stages. If it's a trematode that parasitizes humans, well, usually they spend part of their life cycle in a snail intermediate host. And they're unique in that they can produce surface proteins that mimic the host's surface proteins so that the host's immune system doesn't recognize them. So there are two classes now in the trematodes. One's called monogenia, which means it has no intermediate hosts. The larval stage is an onchomycetium. Or it has a digenetic trematode, which are now called a trematoda. And so that means they do have an intermediate host, or two stages, or two bursts. And two common examples of that are the Chinese liver fluke, Pisthorchis sinensis, and a very common one, Schistosomia, or Schistosoma mansoni, which also causes the disease schistosomiasis, which has snails in their life cycle. So here's an example of Schistosoma mansoni, and so you can see this person is working in rice fields or crops that are grown in water. The mature liver flukes live in the blood vessels of the human intestine. And so once the organism gets in the blood vessels of the human intestine, there are different sized males and females that come together, reproduce, and then they release their fertilized eggs into the feces of the human host. Well, the human host in these situations often excretes their waste out in the field. They don't use latrines or toilets, and they'll often use the human waste as a fertilizer. So the waste gets into the water. The eggs will develop into ciliated larvae. In this case, we call that a myricidium. And then that penetrates the snail intermediate host. Then it continues to go through its development there and releases a motile larvae called a cercaria. And then that swims in the water and looks for a new host, penetrates the skin of this person, gets into their bloodstream, and completes the life cycle all over again. The other platyhelminth that we know is a problem parasite for humans and birds and fish, and a lot of your pets are tapeworms. They have a small anterior head or a scolex that's got hooks in it for burrowing into the wall of an intestine. And so usually the body of this is divided into segments, proglotid segments. They start out small, and as they mature, they get bigger and bigger as we go down the organism until the proglotid segments are mature with reproductive structures and produce fertilized eggs. They get broken off into segments and leave the host body in their feces. This is often a problem with dogs because they like to sniff waste products of other dogs, and that's how they can pick up a lot of these proglotid segments through their nose or through their mouth. Briefly, we want to mention the Syndermata. It's a new phylum. They've combined the acanthocephalans and the rotifers into this new phylum called Syndermata. We just want to look at what rotifers look like because they're an important aquatic animal. They are unique in that they have a complete digestive tract. They have a stomach. They have jaws for crunching up their food. And so usually they're stalked. You find them attached to substrates like leaves. And they have this crown of cilia that creates a vortex sucking in particles that get crunched up by the jaws and pass through their digestive tract. So they have a true alimentary canal. They reproduce by a process called parthenogenesis, which the females produce offspring from unfertilized eggs. Unless there's a stress event, and sometimes they might produce a male that would produce as fertilized resting eggs, but usually it's parthenogenesis. That brings us to the mollusks. There's a real interesting group. The phylum Mollusca includes things that you're familiar with: snails and slugs, clams, octopus, squids. Most are marine, but you'll find some that are freshwater or terrestrial. And some produce a calcium carbonate shell, not all of them, but most are soft-bodied. So here's a nice diagram of the three region body plan of the mollusca. In some way, shape, or form, you'll find mollusks have some kind of a visceral mass as one part. Let's go back here. Have a visceral mass, and then the mantle, and then the foot. So those are your three regions. You'll find other organs contained in these three regions, like your heart, like your nephridium, contained in your visceral mass. They have a very long coiled digestive tract, and uniquely, most of them, not all of them, have this unique grinding structure in their mouth, which has been expanded here, called a radula. It's kind of like teeth on a chainsaw. They rasp it back and forth to rip off the algae or plant material that they're feeding, or in the case of a squid, they use it to rip out the flesh of some fish or other organism that they attach to and are trying to eat. They do have two ventral solid nerve cords. So most mollusks have separate sexes with gonads, but some snails are hermaphroditic. Here's some typical mollusks that we need to be familiar with. Polyplacophorans or chitons. They have many, many plates and a large foot underneath. You'll learn about them in lab. Each of these are plates here, individual plates. And so poly means many. You got 1, 2, 3, 4, 5, 6, 7, 8 plates on the body, but then a big muscular foot underneath. And then you have your typical snails. Gastropoda, some with a land snail with a shell, a sea slug without a shell. Sometimes you'll have a land slug without a shell. And what's unique about gastropods is their body twists through a process called torsion, so that the anal opening comes up right behind the head, usually in this region right in here, so they can excrete its waste. Otherwise, if it was a complete digestive tract and the anal opening was back in here, it would fill up with waste. But slugs go through this unique developmental process where they go through torsion and then they detort after that, and they don't develop a shell. So torsion and detorsion. Bivalves are probably the mollusks that you're most familiar with. They have unique adductor muscles that only function for closing, and the shell is divided into two halves. Some of the bivalves have little sensory tentacles along their edge, or sensory eyes. They're not really eyes, but they sense light. Here's an example here. The mantle cavity of a bivalve contains its gills and are used for feeding as well as gas exchange. We'll look at that here in this diagram, and you'll look at this in lab in more detail. And so, here's your big muscular foot here that they use for moving through sediment. And then you have gills that you use for gas exchange. You have in-current and ex-current siphons where they take in water for filtering. They have an excurrent siphon for discharging that water. They have a mouth. They have a digestive gland. They have a mantle, which in this case is used to secrete the shell. Many of the mollusks use the mantle to secrete the shell. And they have a coelom and a heart that is wrapped around the intestine on the way out. You follow this through. The heart is wrapped around that, and then the anus is on the end of that intestine, and it discharges out the clam when it's open. Cephalopods are the other unique mollusks that you'll probably have encountered. Many of them are predators. They can catch their prey with poison in their saliva or with their tentacles to hold them. The foot is modified into a muscular siphon and part of the tentacles. And in most cases, the shell is reduced or missing, except in the chambered nautiluses. Here are examples of those three. So you see the squid at the top, and the octopus here in the middle. And then here's the one that does have a shell, the chambered nautilus, which you'll see examples of that shell in lab. What's unique about the cephalopods is that they have a closed circulatory system. So later on, when we compare open and closed circulatory system, this is an invertebrate with a closed circulatory system. Not many have that. You can get squid that can be very, very long, up to 10 to 14 meters long, but some are usually around 3/4 of a meter long. Cephalopods have been around a long time, called ammonites. And they were the shelled version and went extinct a long time ago, 65.5 million years ago. Cephalopods have played a major role in human development. These men are sitting on a stack of mussel shells, particular mussel shells from pearl mussels, used to make buttons for shirts around the turn of the century. And so mollusks are probably one of the most endangered animals on the planet. You can see the percentage of endangered organisms that have gone extinct or are in danger of extinction, and you see mollusks makes up most of those. So we'll stop it with that for now, and in the next discussion we'll pick it up with annelids. Thanks for your attention. Hope this was helpful.